In a major scientific breakthrough, a team of South Korean and American scientists have cloned human embryos. There have been similar claims in the past, but this is the first time the work has been fully authenticated. They've already used the embryos to produce stem cells, which in the future could be used to treat incurable conditions like Alzheimer's and Parkinson's disease. Sue Nelson reports.
This is South Korea's self-proclaimed cloning academy. But this time, instead of animals, an international team of scientists created 30 cloned human embryos. They are the first authenticated, fully human clones and were specifically produced to collect embryonic stem cells.
Everybody in our lab did work very hard from early morning till the midnight, and now everybody is very happy because we have first cloned human embryonic cell lines in the world. And now we have to stop and think.
The researchers effectively used the same technique as for Dolly the sheep. And here's one of the egg cells that was used. There were 242 in total, all donated by 16 Korean women. Now this egg cell has had its genetic material removed and replaced by DNA from the same volunteer, a process known as cell nuclear transfer or cloning. 30 of these developed into cloned embryos. They're bunches of cells at this stage before turning into a ball of cells called a blastocyst. And inside here are the embryonic stem cells. Scientists extracted them and then showed that they could develop into useful tissues such as bone, muscle and cartilage. More importantly, these embryonic stem cells are a perfect genetic match. And so, in theory, they could be used to treat the donor for any degenerative conditions, such as Parkinson's, without being rejected. This is therapeutic cloning for medical use as opposed to producing humans. And despite previous claims, such as those from the UFO cult, the aliens, there is no proof of a human cloned baby. But this breakthrough, although greeted by many scientists as a substantial advance, will heighten fears that one day the technology will be used to clone a human being. Sue Nelson, BBC News.
Well, let's talk to Dr. Ian Wilmot. He led the team which successfully cloned Dolly the sheep, and he's in Edinburgh now. Dr. Wilmot, thank you very much for joining us. First of all, how significant is this news in terms of human cloning?
I think it's a very encouraging observation. A number of months ago there was a failure to develop a method in a rhesus monkey, which was by far the closest species to our own to have been tried. And so we were obviously concerned that the similar failure might arise with humans. And it is very encouraging that that appears not to be the case, and they have derived stem cells in this way.
And they've shown that they can work with stem cells, but what does this mean for the treatment of diseases?
Well, I think first we should look to using these cells to study diseases, not to treat them. This is because cloning itself is a rather inefficient process, and many of the cells would have a risk of being abnormal. But we could use this to study human genetic diseases like motor neuron disease, for example, which is not understood and there isn't an effective treatment for it. Rather more than 1,000 people a year die in the United Kingdom because of this condition. So if we could take a cell from a patient with that disease, clone to make an embryo, derive embryo stem cells, and then cause them to differentiate into the type of cells which become abnormal in motor neuron disease, it would give research workers a totally new way of studying that condition.
All right, briefly.
And I think that if we do that...
Sorry, I was going to say briefly, Dr. Wilmot, what's this going to... Yes, I mean, what's this going to mean for scientists who do this sort of research here in the UK?
Well, I think that this is encouragement to those of us who are developing ideas for research of this kind to press ahead and to make applications for the necessary licenses, because we would require a license from the HFEA so that we could begin our own research.
All right, Dr. Wilmot, thank you very much for that.